Thank you for your introduction. I'm Massimo Grillo, and today I'm going to present my work on modeling cerebellar nuclei and implementing nucleocortical pathway. Let's start from a brief introduction about the cerebellum. Here we can see on the left a picture of the anatomical structure of the cerebellum. It is well known that the input from cerebellum are received by mossy fibers, which reach the cerebellar cortex, in particular the granule cell layer, populated by Golgi cells and granule cells, which process the information and convey their inputs toward the Purkinje cells in the Purkinje cell layer. These neurons, in turn, inhibit the activity of the deep cerebellar nuclei, which are the output stage of the cerebellum. In addition to this main pathway, it has been found that there are other pathways. One of them is one connecting the deep cerebellar nuclei toward the granule cell layer. Indeed, on the right, we can see this pathway called nucleocortical pathway, and in particular, there is a section of the cerebellum in which we can see in yellow some lines which represent the axons coming from the deep cerebellar nuclei going to the cerebellar cortex, which are originated by the glycinergic inactive neurons in the deep cerebellar nuclei. So I found out that deep cerebellar models, in silico models in literature, do not implement this pathway. So the aim of the work was to include them in existing models. In order to do that, first of all, I needed to model the DC and glia neurons and then insert them inside the scaffold. About neuron modeling, I used the point neuron model, the extended generalized leak integrated fire, which use these three differential equations in order to model the neural behavior, and some electrophysiological parameters in blue, like the membrane potential, the membrane capacitance, and other optimizable parameters represented in red, which are intrinsic of the model. These parameters can be obtained through an optimization algorithm, which takes the features of the neurons, which are nodal rhythm, linear current frequency relationship, and high spike frequency adaptation. In addition, we need to design a certain stimulation protocol, which consists of the pricing steps, for instance, and target behaviors in terms of fine rates. And then we construct the optimal parameters of the neural model. However, we need to test this neuron in nest and see what happens when we stimulate it. On the top, we can see the stimulation protocol in violet. We can see 10 seconds of rest in which the neurons is not firing. So we can see the nodal rhythm property, and also 3 depolarizing steps with increasing current in which the neurons start firing, followed by an hyperpolarizing steps in which we have no activity. If we focus on this time window and see the graph of the bottom, we can see the oscillation of the membrane potential, in which we can see the interspike interval at the beginning of the stimulation is much more slower than the interspike intervals near the end of the stimulation, meaning that there is a high spike frequency adaptation. If we look also at the graph on the right, we can which we represent the firing rates of the neurons as function of this current stimulation. We can see the no algorithm property when the neurons is not stimulated, the linear current frequency relationship. In addition, we can I also distinguish between the fine rates at the beginning of the stimulation, represented as dots, and the fine rates near the end of the stimulation, represented as squares. And we can see there is a huge difference for different values of the current stimulation, meaning that there is a nice spike frequency adaptation because the fine rate at the beginning is much higher than the fine rate near the end of the stimulation. So now, if we want to include the neurons inside the scaffold, I used an already existing scaffold present in literature, and I included the gliai neurons in the DCN layer, receiving inhibitory inputs from Purkinje cells and excitatory inputs from mossy fibers. These neurons in turn project toward the granule cell layer, in particular contacting Golgi cells. Indeed, looking at the graph on the left, one glycinergic inactive neurons in the DCN layer contacts several Golgi cells in the cerebellar cortex through these red lines. And looking at the functional property of this connection, a glycinergic burst, which lasts around 50 milliseconds, cause a huge suppression in Golgi cells. So to sum up, Thanks to this reconstruction of nucleocortical pathway, there is a new neural population in existing cerebellar scaffolds, and we have a more complex and realistic connectome, which could be very useful when simulating complex cerebellum-driven tasks. So thank you for your attention. I would like to remind my poster session for tomorrow, and I'm here if you have any questions.